name's Amber Fox. This is LAPU BIOL 225, General Microbiology. Today's date is February 20th, 2025, and this lab is the Kirby-Bauer Method. So here I have my most recent fresh broth culture here. I'm going to gently roll between my fingers. It is already at room temperature. And I also have my 2 agar plates here. I'm not sure why, but there's already some organisms growing. This one has more, and then there's a little bit there. So when I do my experiment, I'm going to avoid those areas. And then I have the my swabs here. There's my candle. I have alcohol, and I have my discs here. These two are the blank ones, and then these are the bacitracin or bacitracin and tetracycline, 2 and 2. Okay, so let's get started. So first, well, let me open one of these. So we're going to do 1 disc at a time. I'm going to obtain 1 sterile swab. Get my culture. And I'm going to flame the mouth. Okay. And dip my Q-tip into the broth. Flame the mouth again and close my broth tube. I'm going to set this aside. Okay, so now I'm going to open the lid of my agar, and these are already dated as well. So without touching that, and now I'm going to turn it 90 degrees. And last turn. Ooh, smells. Okay, and then now I'm going to place this in my alcohol disposable jar. Okay, and then I also have my forceps here. These I'm going to place or dip into my alcohol jar. And then I'm going to top off the excess and then just kind of burn off the remaining. Okay. Okay, so now I'm going to obtain my disc while my forceps are drying. Okay, I'm going to grab one. And I'm going to place it on the agar plate. Press down gently. and then back into the alcohol. And let it okay, it's all burned off now. Wait about another 15, 20 seconds. Oop. Ah, okay. Well, I have to relight my flame. There we go. Okay, I'm gonna grab another disc. Or these are just 2 packs, so just the remaining disc here. Okay. I'm gonna place it on my agar. Gently press down. Okay, back into my alcohol. And I'm going to use a blank one now. That'll be the 3rd one, since there are 6 discs. Burn off the excess alcohol. Ooh. I'm afraid of fire, so. Okay. Let this wait a little bit until it cools off. And I'm going to retrieve 1 blank disc. Come on. Okay, and on the agar plate it goes. Pressing there gently and close. All right, back into the alcohol. Okay, so I'm going to repeat this process with the remaining 3 discs. These 2 are the tetracycline and then the 1 blank. So again, let me grab my sterile swab. And my broth, my most recent broth. So I'm going to flame the lid or the mouth of the tube. Okay. Insert my swab. Okay. Reflame the lid or the mouth. And recap my broth tube. All right. And so now I'm gonna inoculate the plate. And tight zigzag. There's a little bit of organism growing on this one too, so I hope, trying to avoid the visible parts. Not sure why I have growth on this one, but in the last section. Okay, and this is going to go in the alcohol. Okay, so now I will grab the tetracycline discs, and I'm going to place both of those as well as the blank one onto this agar plate. I'm afraid of this fire. Okay, I'll let this cool for a little bit. Okay, so now I'm going to grab one and place it on my agar plate. Okay. Dip it back in the alcohol. And drip a little bit. And burn the excess off. Okay. Wait a little bit. Place it on my agar plate. Back into the alcohol jar. Burn off the excess. And lastly, it's the blank sterile disc. Gently press down. This goes in my alcohol. And now I'm going to invert them or flip them over and put them in my incubation area.